The CytoSMART Omni FL is a live cell imager for high-quality whole-well bright field scans or high-throughput dual fluorescence images made inside your cell culture incubator. Monitor your cell culture during large screening experiments in cell growth and viability, transfection efficiency, co-cultures, cytotoxicity assays, and more. To image all your samples in their optimal environment without disturbing your cells, install the Omni FL in your incubator. Make sure the light arc can move freely. Connect the device to the power supply via the adapter and subsequently to the laptop via the USB cable. Place your culture vessel on the Omni FL and position the A1 well of your well plate in the A1 labeled corner of the adapter plate. Keep the long edges of your plate parallel to the long sides of the Omni FL. Open the CytoSMART app and log into your account. The green LED of the device will light up and the camera will move to its ready position. A bright field image of your cell culture will be shown in live view. Select the appropriate culture vessel from the menu. Indicate whether you want to make a whole vessel scan in the bright field channel or go for patterned imaging of all desired wells in bright field and green and red fluorescence. Select the wells you want to include and how many regions of interest per well you want to image. You can move the camera over your culture vessel by clicking on the vessel image on the left side or navigate stepwise with the arrow keys on your keyboard. Adjust the focus level. The autofocus feature provides optimal focus per region of interest in pattern mode, even adapting to changes over time. Set the correct bright field exposure and imaging settings for the fluorescence channels, if applicable. When you're satisfied with the settings, you're ready to start imaging. Assign your experiment to a project and provide an experiment name and description. Analyze your experiment in real time by selecting the appropriate analysis algorithm. Indicate whether you want to image one time point or make a time-lapse. Specify the time-lapse imaging interval and number of time points. Then, you're ready to start your experiment. Monitor the imaging progress on the laptop and check your running experiment anywhere, anytime in the CytoSMART cloud. With the bright field channel map view, you can go from the largest overview to the smallest detail, or take a thorough look at each bright field and fluorescence region of interest individually. Stopping a scanning experiment is not definitive. Append an existing scanning experiment after changing cell culture medium or imaging other vessels in between. After ending your experiment, process your images and analysis in the cloud. Share your interesting projects with your colleagues and export images, videos, or data. Have it all with high throughput, 3-channel imaging with the CytoSMART Omni FL.